Good morning, respected Principal Sir, the Chief Guest of this today's event, Dr. P. MALLIKARJUNA Sir, online and offline attendees and students. A very warm welcome to one and all to the final session of a 3 days hands-on workshop on artificial intelligence in drug discovery and development, a lung cancer case study. Myself, Dr. Antarash Panigrahi, Associate Professor of Raghu College of Pharmacy, hereby welcome you all to this final session of this workshop. I now request our speaker, Mallikarjun Rao sir, to continue the final session, and a lot of things are balanced to learn from his excellence. Sir, now, I now welcome you, sir, to the session.
Okay, for you could be able to sit here for 2 days and another 4 to 4 hours here. Okay, I truly appreciate your patience, your interest, at least uh, not at least keeping me continuously talking. Okay, and because sometimes when I go to some places, when I see students sleeping, I feel like why am I here? But you did not give even that kind of a opportunity. Okay, since the beginning of the this workshop, please make sure that I always say to the people that once it is ready, you let me know. Okay. Um, okay. Can start. Okay. So now coming to this workshop, okay, a day before yesterday, okay, we started this session with theoretical principles of the AI, followed by the virtual screening, okay, where you can see the applications of the AI techniques, okay, in discovering new molecules. Okay, and later on, what we talked about lung cancer, okay, how to choose a disease and how to narrow down our research person. Then yesterday's session, we talked about the genes, okay, then how we can validate the science behind it using a PubMed. Then afterwards, we know that the genes don't work on their own. Okay, they work together with some other genes. Okay, that's why we talked about the strings. Okay, string network. Then afterwards, we talked about the 3D structures of the proteins. Okay, where we talked about the alpha fold. Okay. Uh, then yesterday we have missed one thing that PDB, okay, that is also talks about 3D structure of the protein. Then later on we talked about how the chemistry and biology linked together, okay, because that's where we have seen Campbell and PubChem, okay. Now we are going to see some online tools, okay, which we can use, okay. um, Once you design a chemical molecule, okay, how to predict how it is going to work in a body, okay, prediction, and that's what we are going to cover today. And the first lecture we are going to talk about is PDB. This is a protein data bank. Okay. Actually, without this, nowadays no research happens, especially in drug discovery. Okay, so here it gives access to the experimentally determined 3D structures of biological macromolecules. Okay, whereas yesterday when we talked about alpha field, then we say that here instead of experimentally determined, there you will see predicted 3D molecules. And for drug discovery, it helps convert a validated target into a structured, informed design problem. Okay. Then here I want to say the biological macromolecules means we are referring to the receptors or we are referring to the enzymes. Then here, when I say the structured, informed design, sometimes the people will get confused. Oh, because whenever we draw a chemical, we'll call it as a structure. Okay, so when the people read this, oh, I'm referring to a chemical molecule, but it's not. Okay, in drug discovery, whenever we say structured informed design or structured based drug design, then that structure is referring to the three-dimensional shape of the protein. Okay, that's the important thing that you need to know. The most useful entries are often ligand bound, mutation specific, or high quality domain structures. Okay, this one I already explained yesterday. Ligand means any chemical compound that sits in the binding cavity of a protein is called as a ligand. Okay, so this is a ligand bound structures. Okay, in alpha fold you cannot find the ligand bound structures. Okay, so in alpha fold you will find only the structures of the protein. Okay, but one thing what we need to understand here is, okay, suppose if the molecule is like this, sorry, if the protein structure is like this, okay, suppose if the ligand is going inside, it is going to be flexible, then entire 3D conformation structure is going to be different. Okay, a native protein is like this. Am I right? You look at this one, it is like this. Okay, and you see that these are the things that the ligand forms an interaction. But when you are putting a ligand into it, it may become like this. Okay, so it has to accommodate. That's why in alpha fold you see only these structures because it's not a ligand bond. But here you can see when the ligand bond inside how that structure looks like. Okay, so that is the one. And of course, which EGFR structure should I use? Actually, it depends upon your research person whether you want to target on wild type EGFR or whether you want to target on specific mutant. Okay, and is the structure human? Okay, and is there a ligand in the active site? Okay, and is in a computational drug discovery, we use the word active site and we also use the word binding site. Okay, and the binding site is the one that we use in the case of receptors. Active site is the one that we use in the case of enzymes, but both are one and the same. Okay. Can the structure explain binding, selectivity, or resistance? Okay. Then do not download a structure until you understand what the entry represents. Because this kind of a, what is this called? This kind of a statement um, usually appears in majority of my slides. Why I'm giving so much emphasis is nowadays the people are becoming very less patient. They have less patience. They want to get the things done as quickly as possible. I will tell you a simple example. Nowadays, okay, the people can spend time of 3 hours to see a movie, but they can't even spend even 3 minutes to read something that is related to the science. Okay, it's been happening. Okay, across all. So that's why when the people talk about AI or when the people talk about this one, the people gets an impression that, oh, I need not to do anything. Everything will be done by the computer. But if you go with that kind of a hypothesis, it's very difficult for you to succeed, okay, in your career, research career. Madam, next one. Okay, now, then here what RCSP PDB provides is experimentally determined structures. Here the experiment means they can use X-ray crystallography, or they can use a cryo-electron microscopy, or they can use NMR spectroscopy. Okay, and the cryo-electron microscopy is the latest technique. Okay, suppose if you take the example of COVID-19, okay, that case, okay, when they want to identify the protein, the 3D structures of the proteins that are present in that particular virus, you will get shocked. Okay, within that 1 year, there are more than 6,000 experimentally crystallized protein structures. More than 6,000. Okay, and that is the power of the cryo-electron microscopy. Previously, the people use X-ray crystallography, and X-ray crystallography used to take a very long time, whereas cryo-electron microscopy can do the same work in a much shorter period of time. And very recently, I come to know that there is a new technology called diamond, okay, and which is developed by my own university, Newcastle University, UK, okay, where the crystallization of the proteins, okay, it will be much faster, okay. Just for you to understand, when you are preparing a synthetic chemistry, when you are doing a synthetic chemistry practical, okay, or an organic chemistry practical where you are supposed to synthesize a compound, okay, you know one of the most challenging things is to crystallize that particular molecule, okay. The reason for you crystallize is if it crystallizes, it will be in its most purest form. Then you can see that even that's a small molecule to crystallize, we struggle a lot. Whereas if you say a protein, very big protein, okay, a huge molecular weight, for it to be crystallized, it will take, you can imagine how hard it is. Okay, my own experience, once we thought of doing a neutron diffraction for one natural compound, andrographite, and we want to crystallize it, and we crystallized it for 1 year, 3 months, okay, for the crystal to grow. Okay, so, but now this cryo-electron microscopy is the one that makes things much faster. Okay, the search tools based on the molecule name, sequence, ligand, organism, method, and experimental details. And of course, you can see the interactive 3D visualization. And actually, the RCSP PDB side also, various tutorials. Actually, there are tutorials. Okay, there are very fantastic tutorials, very good for you. And sometimes they also give you the certificate. Okay, structure summary pages. Okay, and identifies whether a drug is structurally tractable. Tractable means whether the structure remains the same throughout, okay, throughout the drug discovery phase, okay, shows the active site, binding pockets, and ligand poses, and supports molecular docking, pharmacophore interpretation, and SAR reasoning, okay. Now here I want to tell something about a molecular docking, okay. A molecular docking is to see whether the compound is going to sit comfortably within the binding pocket of the protein, with the binding of the protein. Okay. So if you think that if you are sitting inside, then it is supposed to be active. But however, for some proteins, we need to inhibit the activity. For some proteins, we have to activate the activity, stimulate the activity. How can we decide that when the compound is sitting inside the binding cavity, whether it is inhibiting or stimulating? Okay, and that one molecular docking won't tell you. Again, it will tell you whether it is sitting inside or not. Okay, that is one thing. Then another thing is molecular docking. What happens means is your protein remains rigid, okay, whereas your ligand you will make it a flexible, so it will go like this, okay. But in real scenario, okay, the proteins will never be rigid like this. They will also move like this, okay. So that's why nowadays molecular docking is also not so successful. So what happens means there is a technique which is called as molecular dynamics. In the molecular dynamics, what happens is this protein also will move, and the receptor also will move continuously, and which gives the more better picture of what happens when the ligand binds within the receptor. Okay. Of course, we know about the pharmacophore interpretation. That means what are the important chemical features that are necessary for the chemical compound to go and sit within the binding pocket of the particular receptor. Okay, and a structure activity relationship is how the chemical uh, groups will modify the bioactivity. Okay, it also explains the resistance. Okay. What does the target looks like when a ligand is bound? This is very important, which alpha fold cannot tell. Okay, alpha fold only tell what the protein is, how it looks like. But when it bonds to the ligand, it won't tell how it is so far. Okay, that's what they are trying now. Okay, the next one. Okay, then this is the simple comparison between the PDB versus alpha fold. Okay, so this is the experimental structure determination. So definitely the confidence values are higher, whereas this is the computational prediction, but still it is working. Okay, and at this point of time, still the people believe this one. Maybe the day will come, then this will dominate. Okay, depending upon the advances in the AI tools. Okay, it includes bound inhibitor, ions, cofactors, or mutations. Okay, whereas no inbound ligand. Okay, because you know that whenever you study a pharmacology or a biochemistry, most of the time you say that this enzyme is active only in the presence of ATP, or this enzyme is active only in the presence of NADP, NADPH. Am I right? So in a PDB, when you crystallize the protein, it also crystallizes along with these cofactors. Okay. So then when the cofactor is there, then there will be change in the conformation. Okay. Whereas at this point of time, alpha fold cannot give you that kind of information. Okay. Then here we talk about the resolution, validation report, electron density, or free. Okay. All these are statistically validated uh, tools to determine the quality of the protein structure. Okay, these are the well-established statistical tools. Whereas here they put some kind of a computational confidence scores. Okay, suppose if you are looking for a binding site, ligand interaction, and docking, still this is the preferable. PDB is preferable. Okay, whereas alpha fold, if you want to know about how the protein looks like, and if the experimental structure is not available, then only we should go for alpha fold. Okay, for time being. And main risk is maybe partial engineered or crystallization bias. Okay, that means the person who did the experiment, if he did it wrong and everything goes wrong, okay, then here may mislead flexible pockets or ligand induced conformations. Okay, it won't tell you because it just predicts. Okay, so that is one of the things. Okay, so use AlphaFold for coverage if you want to understand everything. But if you really want to see a drug discovery point of view, still because our intention is to discover a new molecules that fits into a receptor, still the PDB is the one that works better. Okay, next. Okay, of course, this is the recommended strategy. Then you put EGFR, Chinese domain, Homo sapiens. Okay, here experimental method. Okay, even though there is a NMR spectroscopy, but NMR spectroscopy don't give you the clearer picture. Okay, still the X-ray crystallography and a cryo electron microscope because on the left hand side, when you open the PDB on the left hand side, you will see the different techniques that they use to crystallize the protein. Okay, but the people like us, who are old people, so still we trust only the X-ray diffraction. Okay, but the newer generation is started with a cryo-EM. Then you may ask why. Why experienced people? Sometimes the people think that the people who are experienced, they do not want to try with the new techniques. Okay. One of the reasons is we want to see that how stable the technology is. Okay. X-ray crystallography is known to the scientific community for more than 30 to 40 years. So that gives us a confidence. So if you go for a cryo-electron microscopy, still we do not know that how it works. Okay, because that is called as the repetition. Okay, so again, there should be a balanced view between the older technique and the newer technique. Never ever think that newer technique is always better than the older technique. Okay, but newer technique has some advantages, but the older technique has a repetition. Okay, that's the thing. Okay. Then filter or inspect for ligand presence when doing this one, open the entry and read the title. Okay, is it the, I think it is there in the notes. Next one. Then how to select the right PDB entry, you have to do this one. Okay, select the human, the domain. Even for this one also, okay, because when you look at the protein, okay, later on I will show you the protein that I have worked on. Okay, not easier for the other thing. It's very big, and you will get confused where you need to really look at it. Okay. So then whether it is ligand bond, mutation status, resolution or validation. Okay, next one. Then we also need to look at these kind of things: hydrogen bond interactions, hydrophobic contact, pi interaction, halogen bond, covalent bond. But in what is that called, in PDB, you will find a very less PDB structures where the ligand forms a covalent bond. The reason for that is, okay, when we studied pharmacy, and even the concept was there until 2015, okay, the ligand molecule should not form a covalent bond. If it forms a covalent bond with the receptor, it is stronger. Since it is stronger, it is going to stay in the body for a long time, so it will cause a toxic effect. Okay, that was the thought process. But later on, they say that there are some deadliest diseases in which there are most stronger proteins are involved, so you need to inhibit them permanently. That's why this covalent bond is there. That's why in most of the medicinal chemistry textbooks, still you don't see this word covalent bond when you look at it. Okay, so when you look at this hydrogen bond and hydrophobic bond, most of the people think that the potency of the compound is because of the hydrogen bond. It's not. Okay, most of the potency is because of the hydrophobic contacts. Hydrophobicity is the one that determines how potent the compound is. Okay, then the hydrogen bond, what it will do is, it is the one that determines the selectivity. Okay, actually hydrogen bond is the one that determines which receptor the uh, molecule is going and binding. And this pi interaction and halogen bonds, because if there is a space like this, okay, space, and you want to put a protein ligand, if you put a only small ligand like this, it may come out. It may not stay. What you need to say that you need to occupy it. Suppose if it is occupying like this, then interaction is stronger. Suppose if it is like this, it can come out. Okay. So what in drug discovery, what they try to do is they try to build, completely occupy that space. Okay. And to occupy that space, what they try to do is they will try to take off the pi interactions and very less halogen bond, and mostly it's a hydrophobic one. Because why hydrophobic means is, if you look at any organic compound, okay, you look at the number of carbons are more, and oxygens or nitrogens will be 2 or 3. Am I right? In general, in any molecule, in any drug, in any medicine, if you inspect the structure, and carbon is contributing to the hydrophobicity. Oxygen and nitrogen is contributing to the hydrophilicity or this one. So in any drug molecule, whenever you are synthesizing more things, so we have to concentrate more on the hydrophobicity. That's why CH2, CH, CH double bond, CH2, C triple bond C, actually these play very big role, okay, in terms of this one, but unfortunately, uh, uh, majority of the people do not pay much attention on that hydrophobicity, okay. So interpretation interaction as design tools, okay, next one. So what resolution tells us, okay, because when you open the X-ray crystal structure, okay, then next there is a called a resolution, okay, they will call it as a 3.56, 2.06, 1.08, 0.8. If lesser the value, that is better the PDB quality is. In general, we think that if the resolution is high, am I right? When you look at our phone or computer, if the resolution is high, 640 is lower, 1040 is higher. Am I right? That's what we think. Am I right? That's what really is true. But in the case of a PDB structure, if the resolution value is lower, that means it is better. It is better. Okay. Of course, I don't want to tell the details. Why is it so? Okay. And high resolution means it gives this one. And usually, as a general thumb rule, okay, if the resolution is less than 2 angstrom units, and we consider that it's good, it's good structure that we can proceed further for drug discovery. Okay. And resolution alone is not enough. Of course, there is lots of things of validation metrics and everything is there. And of course, here I want to tell you that actually this is coming for the RCSP PDB, am I right? That is Protein Data Bank from the UK. Then some people they are not happy with this PDB. That's why UK itself, they come up with another institute called as European Bioinformatics Institute. They come up with another database called as ePDB. Okay, so actually ePDB is the one that come up with this validation metrics. So of course these are the validation checks. There is a lot because when you want to synthesize, when you want to crystallize a protein, definitely you cannot crystallize all the things. All the chains you cannot. That's why when you crystallize it, and some of the things are missing because the person who is interpreting, he interpret that, oh, I cannot see anything here. Maybe carbon might be there. So he will do a guesswork. That's why you will see a missing residues and also missing loops. Okay, madam, next one. Next one. 7. Finish. Okay. Can you open this PDB website? Internet. Okay, this is about PDB. Now you understand when you should use AlphaFold, when you should use PDB. Okay, what is the advantage of the PDB and what is the advantage of the AlphaFold? Okay, so now I will show you a few things. Okay, you put RCSP PDB. Yeah. You put yeah. Okay, madam, here there is search button. Okay, actually, those who are interested, just wait a minute. Okay. Here you can see this PDB 101 training courses. These are really fantastic ones. Okay, and actually these are the protein structures. Okay, I don't want to go into details. You see that how complex it is. Am I right? Even small molecule to crystallize will take a very long time, and you can see different types. Okay, is there any shade button? But here, okay, here you put. Okay, uh, yeah, you put that one. Okay, even I also got confused here. Okay, here you put something like a easy FR. Easy FR here. No, no, on the top I see. Go little bit. Yeah, there. Easy FR. Okay, then no need to select anything, just select. Okay, here you can see that there are 673 structures. Okay, and all these 673 structures were experimental. Okay, but you know, this EGFR is present in all these organisms. So we are interested only in Homo sapiens. Madam, click this one. So then it will restrict to only 631 structures. Then go down, madam. Just go down. Okay, wait. Here you can see that X-ray diffraction is a 542, electron microscopy is 47 and 42. Since I say that I have a more confidence on X-ray diffraction, so you click this, madam, X-ray diffraction. Okay, so you got about 542 structures. Go down. Okay, now I told you that if the resolution is less than 2.0, then it is good. Okay, then what I do is I will select 1.0 to 1.5 and also this one, both select. This one also, down. Okay, it is selecting only one. Okay, so in total there are 11 structures. Am I right? In total there are 11 structures. Then I want to see, because the technology is being advanced year by year. So what I want to do is go down here. Okay, I want to see the ones that are introduced, that are this one in the latest years. Then you click this one, 2002. Yeah. So in this is way that how we this one, we started with the 673, then slowly you do it, and this is my way of doing. It doesn't mean that everyone should do in that way. So we need to see this one. Actually, here you can see that EGFR kinase domain in complex with a particular compound. Okay. Whereas in AlphaFold, you cannot find this. You cannot find the information. Go down, madam. And usually this is published in a journal of medicinal chemistry. So since it is an ACS journal, and we are a bit biased towards it. Actually, here you can say that 8A2A, 8A27. Actually, if you see first 2 letters or 3 letters are same, that means it is done by the same experiment or same research team. That's why here you can see that medcam. This is also the medcam. Okay, and the authors are also the same. Can you go down? Go down. So here also the authors are same. Can you go down? Yeah, go down. And of course, here this is one example. These things they are already published in American Chemical Society. Okay, Journal of Medicinal Chemistry. Usually, the people who are interested in drug discovery in academia, for them one of the dream journal is Journal of Medicinal Chemistry. Okay, so I do this one, but this one still to be published, and usually I don't. What I mean is I don't have a confidence on it. Okay, because whoever synthesizes the protein, actually they can put it here, but if they convert into a paper, then I have a confidence. Then definitely I'm going to work on only those 3 compounds. Okay, so here, and can you go up? So now I have 3 compounds, am I right? Then I look at this one. This is 1.43 angstrom units. Can you go down? Here this is 1.01. Can you go down? This is 1.11. So what I try to do is I will go into this one 1.11. Now I click this one 88. This one. Is it okay? So you will get this one. Actually here it can rotate. It can rotate. You can see a lots of things. Okay, here there is a validation report. Here there is a validation report, and if you click that one, it's most important. And the most important thing is just go. Yeah, I think she clicked that one. Actually here, yeah, you see this one is like this one. Okay, can you go down? And here you can choose anything here. There are different this one. Okay, you put this one. So it will take some time to get the things. Okay, and you can play with it, and it is going to take a very long time. Okay, so it will have this kind of a ones. Okay, and when you see the structure, it is very good, but you need to know what is this rubber. Okay, what is this loop? Okay, what is this? Actually, you you need to know about it. It looks very nice, but if you do not know the science, it's very difficult to find it. Okay, so you can like this, you can play with it. Okay, then slowly you can adjust to it. Okay. So that is about the PDB. Okay, the next one is I'm gonna go to Swiss similarity. Okay, again recap whatever we discussed. Gene, okay, gene network. Okay, 3D structures of the proteins. Okay, one is the predicted ones, another one is the experimentally determined one. Then the association between the chemistry and biological activity data, that is Campbell and also PubChem. Now we are going to look at um, uh, because whenever we design a molecules, we want to design a molecules which are new and which should be better than that one. Okay. Suppose if you look at, I think you all know about the cholinergic drugs and adrenergic drugs, am I right? They are there for a very long time, very long time back, am I right? At that time, there are no computational models. At that time, there are no 3D structures of the proteins. There are no 3D structures of the proteins, but still they could able to discover lots of medicines. Okay, even the adrenergic itself. Okay, that's why you see adrenaline, histamine, and acetylcholine. Okay, of course, dopamine and all these things are there, but these three are, because why I'm saying this one is the importance of the chemistry in drug discovery. Okay, so they designed all the molecules, cholinergic drugs, adrenergic drugs, and those which are acting on the nicotinic receptors, everything. Okay, even respiratory system drugs are developed based only on these chemical structures. Okay, by modifying the structures of the noradrenaline, by modifying the structures of the chemical structure of the acetylcholine, by modifying the chemical structure of histamine, they could able to come up with many types of medicines. Okay, you imagine after introduction of so many things, no better adrenergic drug came until now in the market. Okay, because those days science is very strong. The people are very strong in science. Okay, they don't simply depend upon these AI tools, these computational tools, and all those things. They really thought about the acetylcholine. Okay, oh, why it is working, how it works. Then you can see that even they got the selectivity. Muscarinic receptors versus nicotinic receptor, they got the selectivity. Even if you look at the adrenergic, beta 1 versus beta 2. Okay, but at that time there are no crystal structures. Okay, because those who are interested in drug discovery, you always need to know the past, you always need to know the history. Okay, so without having the protein structure, okay, only by knowing the chemical structures of the neurotransmitters, they could able to develop the fantastic molecules which are still in the market, which are not yet being withdrawn. But the newer molecules that are being developed, after 2 years or 3 years, they say that problem, this problem, kidney problem, that problem, this problem, they will be withdrawn. Okay, that's why our body produces a lot of chemistry, and we need to know, understand that chemistry. From that we can see that, and that's what we call it as ligand based drug design. Ligand based drug design, that's what that's what we call it as a medicinal chemistry. Okay, so if you know the ligand molecule, and if you want to know the similar molecules, okay, then one of the things that we can use is a Swiss similarity. So Then you may ask me now, Arjun, previously also you talked about chemistry. Now you are talking also about chemistry. What is the difference between these two? Then you have to see that now you are talking about how to generate the new molecules, new ideas. Okay. Then Swiss Similarity is a browser-based platform for ligand-based virtual screening. Okay, it searches chemical libraries for molecules similar to the query molecules because it's very easy. Okay, it will tell you that from which company you can buy and everything. Okay, it supports early hit finding, analog search, repurposing ideas, and scaffold exploration. Okay, and here I need to emphasize that the meaning of the hit here is not exactly the same meaning of the hit that you will see in a medicinal chemistry textbooks. Okay, because that is a different. Here, hit means okay. I have a 10,000 compounds. Out of 10,000 compounds, how many compounds are going to be drug-like? That is not. That means straight away you are getting it done. Okay, so please don't get confused with this particular term. Okay, this is analog search. Analog search means just now I told you that in a molecule, first they'll try to find whether anything is sitting or not. Then afterwards, they will try to increase the size of the molecules. Usually, how they increase the size of the molecule is by using only CH. CH, it could be CH2, it could be CH double bond CH, it could be C triple bond C. By that one, they try to expand. They try to expand. Okay. So that one, okay, and use similarity results as a evidence informed hypothesis, not as a final activity proof. That means you can use this similarity when you are putting up a research proposal. Okay, and then you can understand the 2D, 3D, and a combined similarity methods. Okay, how we can compare these two structures of the similarity? Sorry, how we can compare the similarity between the two structures? Many people think that by observing with eyes we can do it. It's not at all possible. Okay, there is something called the Tanimoto coefficient. Okay, there is something called as a 2D fingerprints. Another one is called as a 3D fingerprints. Okay, there are a lot of newer techniques out there. Okay, here one thing what you need to understand is, okay, when you are looking at the isomers, I told you that the positive plus isomer may be active, minus isomer may not be negative, but it's exactly the same molecule. Okay, so if you see the chemical structure, they look exactly the same. Am I right? If you write the canonical smiles also, it is exactly looks like the same. But what is the difference is that 3D shape of the molecule is different. In plus, it may be upside, and in minus isomer, it may be downside. So that is called as the shape complementarity. Okay, that's why nowadays the people will call it as the shape-based virtual screening. That means if the compound is like this, so your molecule should be like this. But if your molecule, if your compound is like, if your receptor is like this, then your compound should be like this. Okay, so the shape similarity. Okay, so that's what we can use this one. Okay, RCS Swiss similarity. Okay, then of course from this you will go into the Ekman properties, that is Swiss ADME. But can you go to the next one? Yeah. Then okay, can we identify a molecule chemically related to a known active compound? The reason for that is because instead of synthesizing a completely new molecule without knowing that whether it is going to be active or not, okay, generally we believe that if the chemical compounds are structurally similar, okay, they may produce similar biological effects. Okay, of course, from that only the hypothesis came like a bioisosterism. Okay, even in my first lecture, I have shown you the four structures of these EGFR kinase inhibitors. If you look at those chemical structures, they look very similar. They look very similar, at least when you see them. Okay. Okay, can we find analogs, approved drugs, bioactives, or purchasable compounds for follow-up? Okay, here I want to spend some time here. Okay, and suppose if there is a approved drugs, that is a drug bank. Okay, then if you are finding a new use of the existing drug, existing medicine, life is very easy. You need not to do the toxicity, you need not to do the safety profile, because it is already in the medicine. Okay, people are using it. That is a very shortcut method, very effective method, efficient method. Okay, so that's the approved drugs. And actually, you have seen that yesterday you have seen that Campbell library. Okay, so apart from Campbell library, there are also a lot of chemical libraries are there. Then what is the usefulness of this Campbell library is at least those compounds have shown some positive biological effect. Am I right? At least they might have shown some kind of a safety profile. So instead of searching for a databases which we do not know whether they are going to be biologically active or not, so it's better to go and look at the Campbell database, okay? Because the activities may not be the activity that we are looking for, but at least they are biologically active. So definitely the chances will be higher to get to this one. So that is one thing. That is called bioactives. Then we go for a purchasable compounds. Okay. For example, zinc library. Because if you want to do this starting synthesis from the beginning, it is going to take a long time. Why should we waste money? Why should we waste time? So look for the purchasable compounds. Then you directly purchase from them. Okay. Always remember, drug discovery doesn't mean that finding some compound which is completely new. Okay. Drug discovery means finding a compound which can treat a particular disease, whether it is a known compound or a not known compound, it doesn't matter. Okay, so sometimes we are so biased towards the chemistry, so we think that drug discovery means it must be completely new chemical compound. Actually, it is a waste of efforts. Okay, then we can do the structure activity relationship. Okay, can we prioritize hits? Again, I told you the definition of the hit is different from what you will see in a medical chemistry textbooks. Okay, of course, these are the things that we can do it. Next. Of course, the core concept, ligand-based virtual screening assumes that chemically similar molecules may show similar biological activity, but it may not be true. Okay, but you need to do little bit tweaks here and there. And Swiss similarity compares the query molecule against a selected chemical libraries. That is biggest advantage. That is really the biggest advantage of this one. Okay, and this Swiss similarity, because I told you that PDB and EBI is from UK. This is from Europe. That's why all the drug discovery is happening only in those parts of the world. Okay, that's why sometimes from the Asian region, when we are saying that when we are talking about a drug discovery, the people from that part of the world they may not appreciate much. The reason for that is if you look at the Pfizer itself, per day they will synthesize 200,000 compounds. Per day they will synthesize 200,000 compounds. Okay, so we are no way compete with them. Okay, so that's their concept, huh? But it doesn't mean that we can't do it, but we can do it. But the technology that they have, the infrastructure that they have, the manpower that they have is much higher. Okay, so that's why those who are really in this class, those who are really interested in drug discovery, okay, you have only two venues. One is UK, the another one is Europe. Okay, those two are the things. Okay, of course, if you are in a formulation one, definitely India is the best. So then it ranks the output molecule by similarity score. Then of course you can do this one. Okay, here these are the important ones. Similarity is not a potency. Okay, that means if high similar means it may be highly active. You cannot assume that. Similarity is not a selectivity. You cannot say that. Okay, similarity doesn't mean that it is a safe. Okay, high score only increases the probability that the molecule may increase. It's always the probability. It's always the probability. Okay, here also I want to say something. Okay, it's a nature. Actually, it's what the physical chemistry says about the entropy and the enthalpy. Am I right? Which we never talk about in the drug discovery. But entropy and enthalpy are the fundamental uh, forces behind all these molecules to go and sit into the receptor and produces a drug discovery. Okay, produces a biological response. Am I right? So, what this entropy and enthalpy says that, in simple words, you cannot find anything 100% perfect. Cannot. That's why you cannot find anything 100% pure compound. Okay. That's why I always tell to in my class, I always tell to my students, if you find a label 100% pure, that means you must not buy that product. It's not at all possible. In a similar way, we cannot find any person with 100% good qualities. It's highly impossible. Highly impossible. Okay. So that's why, the physical chemistry concepts like entropy and enthalpy, they are the fundamental theoretical principles. Even the hydrogen bond, just you ask why the hydrogen bond is forming. You say that oxygen and hydrogen. Then you say, oh, what is so great with the oxygen? Then again you have to go to the entropy and enthalpy. Everything goes to the entropy and enthalpy, and those two are and the third one is the free energy. These three are the fundamental properties of any molecules going and sitting into the drug discovery. But unfortunately, what happens means we study physical chemistry separate, organic chemistry separate, and medicinal chemistry separate. That's why we don't understand how to connect those two. Okay. So, okay, that's why it's only the probability. You can say the 99%, you can say 98%, or you can say 96%. Right? Suppose if you say one person is good, that means that person has relatively more good qualities than the bad. Okay, bad means okay, that person has relatively more bad qualities than good. Okay, that's because we need to understand that. Okay, next level. Now, of course, you can put this correct molecule, canonical smiles, library choice, method choice. Okay, the project objective. It entirely depends upon it. Okay, and of course, these are the why it matters. Okay, of course, you can use the different methods. Okay, here, of course, you can. Yesterday, when I talked about PubChem, I told you that Campbell is better than the PubChem when it comes to the drug discovery. But when you are copying the smiles, okay, always better to copy from the PubChem. Because somehow the American Chemical Society is very strong in chemistry. Very strong in chemistry. If you do a small mistake also, they will get very angry. That is America. America is only on chemistry, whereas UK is more on biology. More on biology. So you need to know that. Okay? So of course, a library choice, you can use a drug bank, or you can use Campbell, or you can use Jet database. Okay, define mutant selectivity, oral profile, or CNS goal first. Okay, depending upon your goal. And of course, this is the beginner workflow. Okay, whatever you want, you can do it. Okay, and of course, these are the ECFP4 method. Okay, these are the chemical similarity methods. Okay, of course, that itself will take a very long time. But those who wants to understand what is this chemical similarity methods, go to the PubChem. In the PubChem, there are lots of tutorials on chemical similarity. Actually, to be honest, I tried my level best to understand what it is, but I could not able to complete the course at any time. Okay, because since you are young, actually, you people need to spend the time. Okay, here also I need to say one thing. I also tell you this one. Am I right? You are still young. Am I right? You are going to build the career for another 40 to 50 years. Okay, what is Newton's third law says? Action and reaction are equal and opposite. So if your basic foundation is strong, and if you kick that foundation strongly, you can jump to the higher levels. But if your foundation is not strong, if you kick it, then you will fall down and you will break your knee. So that is the difference. Why you need to build this structure. Okay, here also I want to say one more thing, just one thing. I always tell to the people, sometimes if you want to enjoy your life, you can enjoy your life at any point of time. Even at the age of 60 also you can enjoy. It's not that you should enjoy only during this time. Okay, so this age you should make use of to make the basics much stronger. Okay, the next one. These are the various databases, approved drugs, clinical candidates, bioactive molecules. Of course, this is the commercial spaces. As I told you that if you go from here, it's easy. Okay, you can get something. Okay, very fast. Of course, the clinical candidates means that means they have failed in a clinical trials. But they have done lots of experiments, preclinical and everything, am I right? So some data is there already about those molecules. So then you go for the clinical candidates. Of course, you can use actually here generative AI. You can say, hey, can you tell me where I can find the approved drugs, or how I can download the approved drugs, okay, or which website I should look for? It will tell you. In that way, you can help. Actually, nowadays, as a lecturer, actually, we need not to tell you where you search for, okay, because you can ask generative AI. What we need to tell you is the concepts, basic concepts we need to tell. And you also should not ask us, oh, Arjun, can you tell me where can I get that information? Where you need not to ask me. Just go and search in the generative AI. It will tell you. That is the purpose of the generative AI, but not the purpose of replacing the science. That's the key concept. Okay, then you also look at this bioactive molecules. Okay, then this zinc database, which is very powerful database, and this zinc database is also from the American Chemical Society. Okay, I think Stanford University. Then of course you can use large virtual space. Unfortunately, what is happening is the people are going directly here. The people who are maybe very anxious about the drug discovery are more enthusiastic about drug discovery without understanding the concept. Directly they will go there and they will fail. Of course, once upon a time, I was also belong to that category. That's why the experience tells us a lot. Okay, next one. Then, of course, you can use the 2D fingerprints, 3D methods, pharmacophores are combined. Okay, but no, each and every method has its strength and also it has its limitations. Okay, one clear thing that you will see in my slides is whatever I'm talking, whatever the tool that I'm talking about, whatever the database that I'm talking about, I'm talking about the strength and also I'm talking about the weakness. Okay, this is called as unbiased. In a research, we should be unbiased. Okay, we need to understand what are the strengths, and at the same time, we need to acknowledge what are the weaknesses. So, strength means it helps us actually to do. Weaknesses, it will makes us to alert. Okay, so that is the main reason why in most of my slides, I always try to put strengths and weaknesses. Okay, so of course, there are methods you can combine all these things. Next one. I think this is the last. Okay, high similarity score means it is very close to the query molecules. It doesn't mean that it is there. Okay, it may preserve the same scaffold, but still you need to do the docking and all those things or experimental methods. Okay, and my own experience again, I go to 0.7. Okay, always I go to for anything similarity, I also go for 0.7. Okay, actually, when I started this one serious about it, since I'm very energetic and enthusiastic and not that much mature, always I go for 0.9 above. Failure, 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 failure. Come down 0.8, also didn't work for me. Then 0.7, I'm quite comfortable with it. Okay. Of course, there are people who can work with 0.5 also. Okay. Then you can, because you may ask me, Arjun, can you tell me why you can't go for 0.9? Yes, I can go for 0.9, but to synthesize that molecule, I need to have $1 million. Where I can get that funding? So we always need to look at how much funding is available to me. According to that, what I can do, only you do that. Okay. You may have a lots of ambitions, but you don't have a infrastructure support and you don't have a financial support, you cannot do anything else. Okay. That's why it is always a balance. Okay. Whenever you discover a molecule, we always say that it's a compromise between the safety and toxicity. Am I right? Sorry, potency versus safety. Here also, whenever you want to do research, it's a compromise between your ambition and the support system that you have. It is always the difference between those two. Okay. So then, okay, next one. Yeah, chemical interpretation. Does the hit shape the same pore scaffold as the query? Okay, does it preserve key hydrogen bond donor? These are the questions that we will ask. That means when we are comparing the molecules. But drug discovery interpretation is this, is what does it mean? Okay, so is the hit already approved, clinical or bioactive? Is the molecule is too large or too less? Okay, whether that compound is reported to exhibit EGFR activity or not, or can we dock that molecule into the EGFR kinase domain or not, or whether it's helpful to work on the mutations. Okay, next one. Finish it. Okay. Then can you open that Swiss similarity? Actually, I put similarity settings. Yeah. Here also you can see there are tutorials for everything. There is a tutorials. Am I right? Here you can see. Okay. Wait. Okay. This is the difference between. I think I need to be a bit cautious. I think this is the observation. This is the difference that I have observed between UK, Europe, and somewhat US with the Asian ones. You know, what they do is they make everything publicly available to all over the world. All over the world. Because what I told you, for to make a drug discovery, because it requires some money, the government will fund that money. That money government spends to us is paid through the tax. Okay, so then whatever the research that we do, we have to give it back to the community. This is how they will give it back to the community. Whereas in our parts of the world, we try to keep everything to us, whereas they are keeping it open to everyone. Okay, and they don't see any competition. This is called as a pure drug discovery board. Okay, here you can do everything. You can do the docking, you can do the parametric optimization, you can do the side chain, you can do the bioisosteer, you can do the target prediction. Okay, you can do the ADME, you can do the Swiss similarity, everything. Okay, so here I have everything, but at the same time I use some softwares, okay, which are being used by the pharma companies, which I have to pay about around $200,000 every year. Okay, but when I compare the output from there to here, it's almost the same. It's almost the same. But sometimes these works much better, much better. Okay, because many people use it. So these are also the AI tools. If you ask how this side chain is working, how this bioisosteer is working, how this target prediction is working, all these are like that. Okay. Of course, so now we are talking only about a Swiss similarity. When you click this here, here you can see a tutorials. Okay, we click that. Yeah. So you get all the tutorials and really the fantastic ones, and you will get it. Okay. Can you go to the Swiss similarity? Here, come down. Here you can say put it in a files format. Okay. Or suppose if you do not know, if you want to draw a molecule, you can use this one. Can you click this one? Okay. Actually, this one, okay, a software called Marvin. It is from Handson. Okay. Many people say that, oh, I cannot use Kendra because I need to buy and all those things. Whereas Handson is free for academics and one of the best chemistry drawing tools. Okay. Previously to this one, there is something called JMOL, but now they moved it to this one. Okay, can you go back? Can you, yeah, okay. So here you can select a class of compounds here. Can you click it? Okay, here you can select drugs or bioactives or commercial, anything. Okay, now here you can see this Campbell approved drugs, Campbell drug candidates in clinics and drug bank. Actually here, all these are the similarity methods. These are the 2D similarity methods. This is the 3D similarity methods, and this is 2D and 3D together. And usually what we do is we need to go one by one, and you need to really look at this one. So it will tell you, it makes the things much easier. It makes the things much easier. Okay. So yeah. That's all. Okay. That is about this Swiss similarity. Of course, you can start screening. What will happen means it will take some time. Okay. Of course, if I'm not mistaken, you can also create an account. If you create an account, actually, it will come to your email box. Or otherwise, once you put it, depending upon your query, it may take about, sometimes it may take about 1 day, 2 days, 1 week. It depends upon how busy the server is. Okay, but here we need to appreciate their kind of a, what is it called, support for drug testing. Madam, next one. Swiss ADD. Okay, that is about Swiss similarity. Okay. Now, what we have seen is we have seen how to find the similar compounds, right? Then after seeing the similar compounds, what we need to see that whether they are going to be safe or not. Okay. So before going to the experiment, you look at your ADME properties. Here I want to say one thing, all right? If your computer prediction says it is toxic, if you do the experiment, definitely it will be toxic. If your computer prediction says not toxic, but we do not know your experiment can be non-toxic or toxic. Okay? So then even though what is the limitation, or I say what is the strength, at least it will tell you what are the toxic compounds. Take out. Okay, that one you should not really think about. Definitely they are going to be toxic. But whatever it says non-toxic, and those things you have to go to the experiment, then only you have to confirm that whether they're really toxic or not. Okay, that is one thing. Then here, okay, many potent molecules fail because they are not developable as drugs because kidney cardiotoxicity, liver toxicity, lots of toxicities. Okay, and unfortunately, nowadays the people have started believing that the toxicity is because of some of the mutations in the some of the people. Okay, especially for Caucasians, CYP3A4 is very predominant. CYP3A4 is predominant. I already told you, I think if I'm not mistaken, to you only I told that Europe and those countries, they will drink beer almost 5 liters, but nothing will happen to them. And sometimes what happens, we blindly copy them. They are drinking, nothing happens, if I drink one. So, because their body structure, that genes that they have, they can protect them. Okay, that's the thing. Okay. So then Swiss Academy gives a fast, free, browser-based first look at the drug likeness and pharmacokinetic plans. The tool is especially useful before synthesis, purchase, or wet lab prioritization. Okay, that means you need, because this is a free tool, right? So you can use it. Okay, you need to spend only your time. That's all. Okay, and helps convert a chemical structure into a developability questions. Next one. Now here what you can do is you can calculate all the physicochemical descriptors. Okay. Of course, if you want to follow the Lipinski rule of 5, you follow the Lipinski rule of 5. But I strongly suggest you all. Okay. For examination purpose, you remember Lipinski rule. But afterwards completely forget about it. Okay. Then the other thing, you need to be very careful with it. Until now, the chemical domain is the one that is dominating the drug discovery. But now biological molecules are coming. Antibodies, okay, monoclonal antibodies, lots of things are coming. So you need to be really aware of, because we are going to live in that space for another at least a minimum of 50 years. Okay, just now someone asked me that, okay, how should I prepare myself from now? Always think what is going to happen in the next 5 years or after 5 years. Don't live in the present, live in the future. Okay, so how to get that? Maybe if you Google it, actually you will know, or actually you can ask generative AI. Okay, in 2026, this is the drug discovery. What do you think? What is going to happen in 2030? Then it will tell you. Because it is going to do all the things, right? All the current information, all those things from the web tools, whatever is being happening, then it will tell you. Then you can ask, well, I want to pursue my career in drug discovery. I completed a pharmacy course. So what kind of skills do you think that I should gain? Okay, for me to be eligible to work in a drug discovery by 2030. It will tell. That's where you need to use. And those kind of sometimes advices we also cannot give, okay, because our knowledge is much limited, whereas your ChatGPT can help a lot. Okay? And actually, ChatGPT, not ChatGPT, it also can help a lot, because sometimes I ask, how should I save my income tax? It gives just fantastic ones, fantastic ones. Okay, then lots of things it will tell you actually. Yeah. So then physicochemical descriptors such as molecular weight, TPSA, hydrogen bond donors, and all those, lipophilicity and water solubility, and of course, all these are the various drug likeness filters. Okay, I'm not going into that one, but these are only the filters. Okay, of course, nowadays no one is following this. And these are the ADME outputs, JA absorption, blood-brain barrier permeation. And one of the things that actually, even now, most of the textbook chemistries, sorry, medicinal chemistry textbooks do not talk about is efflux pumps. Okay, they talk very little about it. CYP inhibition also they will touch and go, but there is a lot about CYP inhibition. Okay, of course, these things you can take the ChatGPT's help. You can take the ChatGPT. When I'm saying ChatGPT, don't think that I'm referring to ChatGPT. Okay, I'm referring to the generative AI. Okay, don't go and say that, oh, Archie says ChatGPT always, so ChatGPT is so good. Please don't say that. I'm not here to say that ChatGPT is the best. Okay, so then these are the visual outputs. This is a good thing, because that's why what the people say that a diagram, a diagram talks more than what we can speak or what we can write. A diagram is the one that speaks about 1,000 words. Okay, that's why they will try to put all these things into a visual picture, into a visual picture. Okay, that's why you know that we have the 5 main organs in our body. If you go to movie, you know why we enjoy the movie, why we remember movie for a long. Because while this one your eyes is working, okay, your ears are working, sometimes you will laugh, this is also working. Okay, that's why for your brain, it is going through 3 different routes. Okay, that's why you will retain it for a longer period of time. That's why most of the tools, okay, that's why actually that is also one of the reasons why I try my level best to make sure that my slides are looks like a bit acceptable to see. The reason for that is that creates an impression. Actually, the same thing also we can simply type and show it in a simple Word document. So that, because in a drug discovery that is also very important. Okay, because you have to go and defend to someone. So your visual thing is very important, for which the Microsoft PowerPoint is the best. Okay, you can do lots of things in PowerPoint. You can create a lot of diagrams. Okay, even ChemDraw, you can draw a powerful diagrams. Okay, of course there are a lot of AI tools are there. Okay, still I make use of the AI tools, but for me to give the idea, but I never use that into actual drawing and image. Okay. Once upon a time, I used to draw I used to do everything on my own. I am capable of doing everything in this format on my own, simply using PowerPoint. Okay. But those days, it may take about maybe 2 hours. But now with the help of the ChatGPT or something else, I can do it only in 2 minutes of time. That is the difference. Okay. So just using this one is early stage triage tool. Okay. Here you can see the triage tool means comparing more than one property. Okay, so don't listen to one. Look at the overall picture, then make a decision. Okay, that is meant by this one. Okay, then it supports the decision about which molecules deserve paper docking, experimental ADME, and wet lab testing. Of course, as I told you, Swiss ADME says the toxic, forget about it. If it says not toxic, then you have to further investigate. You cannot simply believe it. Okay, of course, this is one known drug comparison, benchmark approved EGFR inhibitors. Okay, then it that means you can put this EGFR drugs. You look at what is their admit profile. Okay, don't follow any Lipinski rule of five, don't follow anything. You put them to see the admit properties, then you put your molecule, then compare the admit properties. Then if they are similar to it, that means it is good. Okay, so that means that is called known drug comparison. Then screen large hits from similarity switch. Okay, then of course lead optimization, identify proper liabilities. Okay, and CNS focused projects, because this is one of the very difficult things. Okay, because for some we say CNS activity is not good. For some drugs we say CNS activity is good. Okay, of course report preparation. Next one. Of course, these are the some of the questions that you should ask. What should be the molecular weight? Whether it should pass the Lipinski rule of five, whether GA absorption is high or not, whether the blood-brain barrier is permeant or not. Of course, these are some of the expert questions that we do. Okay, so here also you will see these kind of things. What does it mean? Means here when I'm preparing this one, actually these are the things that research student is the one who is supposed to do, and this is the one what the research supervisor is supposed to do. Okay, because we need to define what is the supervisor's role and we need to define what is the student's role. So whenever you see the beginner question, that is what the student has to do. Okay, and what the expert one is, what the research supervisor is the one who is supposed to do. Okay, so yeah, then if you say that yeah, Swiss ADME interpretation depends on the intended. Yeah, I think yesterday I said that there is a one phrase that drug discovery people use. Now I got it. Kill fast, fail cheap. Kill fast. That means you kill the drug discovery project at the earliest possible, so that you can save lots of money. Okay, so in drug discovery, all these Pfizer companies and all those things, what you can see is that some people they say that big companies they are somewhat they do not have any emotions or anything, so they can fight the people immediately without any notice. The reason for that is, suppose if I'm working on a drug discovery project, I develop some love towards it. Whatever the people say that since I love it, I say that oh, it's best, best, best. There is no way I can come up. And when I'm trying to say best, 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 I'm asking more and more money. And the other person knows that definitely this is not going to work. Then what they say, they'll kick me away. Then someone will come and they will close it. They will close the project. And closing a drug discovery project is one of the most difficult things. Okay, even when I moved from my cancer research to the other, it is one of the most difficult decisions that I have made. But that is one that I have to make, because otherwise I cannot progress. Okay, especially knowing that whatever the cancer research work that I'm doing is not to the level of what UK is doing, what US is doing, what Europe is doing. Okay, then I also tell you why I take the infectious disease. Somehow they don't do it because they don't have infectious diseases there. That's why I'm doing it. So when I do it, I will get some kind of a recognition in my own field. So you need to play around, and sometimes you need to make a very hard decisions about your path in your career. Okay? So you always, because we always say that ligand must be flexible, am I right? To fit it into the receptor. In a similar way, in our career, we also must be flexible. You cannot say that I studied pharmacy, so I am going to be a pharmacist. Oh, I am a medicinal chemist, so I only do medicinal chemist. It doesn't work anymore. Okay? If you are flexible, you will go in. And you must be competitive. Because the people will say that receptor and protein or drug is a competitive binding, am I right? So nowadays career is also competitive. You always compete with someone. Okay? Then only you will go to a better ones. Next one. Okay, best input is again you put the canonical smiles. Okay, paste one molecule per line, and Swiss ADB, what you can do is If I'm not mistaken, if you put 100 molecules, you put 100 molecules into it. But here, I do not know, but still, I like that Swiss ones. Okay, I think they are professional and ethical. Some people think that when we put it in a public domain, someone may catch our thought process. But still, they know actually who is doing what. But actually, um, these are highly professional ones. That's why in a research, the people use two words: ethical and responsible. Ethical and responsible, and all these databases actually the ones that reflect all those things. It says that how professional they are, how ethical they are, how responsible they are towards the community. Okay, that's why they are sharing all those databases with everyone in the world. Okay, at a free of cost. So use the parent compound unless the salt form is specifically required. That means if you find a salt, kick out the salt. Okay. Then what to document? These are the things that you need to document. Okay. I already told you, you always use a plain text file. Never ever use Microsoft Word. Okay. Whenever you are copying and pasting smiles and all those things, always put it into a notepad. That is the safest way of doing it because of the way that MS Word is developed. Okay. So next one. Then these are the things that you will get, whether it is absorbed or not, whether it can pass through the blood-brain barrier or not, whether it is a substrate for the PGP so that it will be effluxed out or not, whether it is going to cause CYP inhibition or CYP induction, okay, and the skin permeation. Can you tell me what is the importance of CYP inhibition in drug discovery? CYP modulation, what is its importance? Okay. In most of the textbook, what we say is that it talks about the metabolism, but in drug discovery context, it talks about the possible drug-drug interactions. Because when we put that one, okay, when we take a two drugs, one drug may induce CYP, so it will metabolize the other drug than that one, so it affects. So in drug discovery, CYP studies are used to determine the possible drug-drug interactions. Drug-drug interactions. Okay, that one what we need to see. Okay, of course, the metabolism is the one. Once it comes into the market, that the people will talk about the metabolism. Okay, of course, the skin permeation. If you are looking for the transdermal ones. Okay, next. Yeah, can you go to this one? Here you can see that DDI, that is a drug-drug interaction. Next. Then of course there is a boiled egg model, okay, because different people will come with different things to make it attractive. So there is a white region, yolk region, and outside region. Actually, when you put some molecule, you will get all those things. Okay. Then it's always better that if it is in the white region, then it means it is going to be orally bioavailable. Okay. Then if it is a fall in the oil region, possibly it may have some kind of a CNS activity. Suppose if it is outside, then it gives you some kind of a visual piece. Okay. Next. Then what we need to do is we need to inspect all these things. Okay. And how we interpret is the pink region represents the preferred space. Okay. Out of zone identifies there are some opportunities you can do it, and always the key point is do not optimize one axis. You look at all the properties together. Next. And the useful outputs, okay, pains, okay. Actually, with a due respect, even I have, when I did my PhD in natural products. I'm a big fan of the natural products. Okay, I continued my research. Okay, so then after some time, I asked myself, how many of you know about curcumin? Is it really fantastic molecule? Yes or no? How come why pharma industry does not have a brain? Why they are not converting into a tablet? Why they do not have a brain? No, tell me. No, no. Okay. That I will tell you. You are not taking curcumin. You are taking turmeric. I will tell you that one later. Where, where our biggest scientists made a thing. Okay. Because one day I got this kind of a thought. Curcumin is best. Everyone is saying it. And actually, one day I did a search. Curcumin does not work for what disease? Everything in the world. Then I thought that why the pharma industry are fools or what? Then this one in drug discovery, you need to be very careful. We need to be very, very careful. Okay. Then I developed a hypothesis that it doesn't work. Of course, my own experience also says that I worked on the gingerol, that is a ginger, am I right? Of course, in Malaysia, the people call me as ginger boy. Okay, and that is my favorite ones. Okay, and I started my research from there. Then one night, suddenly I got this kind of a thought: why pharma industry is not working on the curcumin? Okay, because they can make it millions of dollars, and they cannot do it, so it's not. Okay, now coming back to the question. Okay, your question I will tell you. This is where all the biggest scientists made a biggest mistake. Okay, this is my only hypothesis, sir. Can anyone tell here? Paracetamol, why it should be in a formulation form? Why can't you take a paracetamol like a glucose and eat like this? Why?
Higher doses injurious to liver.
Even you take small pinch also can take it, right? Okay. So that means we cannot take any medicine in its pure form. It should be in a formulation form for so many reasons what we study in pharmacology, pharmaceutical analysis, pharmaceutics. This is where the turmeric powder itself is a formulation. So when you take that entire turmeric, it's a formulation. So if you take it, it works. But when you take out the curcumin out, it doesn't work. Okay, that's where all the people they made it. Sometimes I scold myself. How come I could not able to understand? This is my own hypothesis. Okay, not someone told me. This is what I say. So if you take the entire vegetable, am I right? It works. Am I right? So you imagine we all are here. Am I right? Very fantastic. B complex tablets, everything is there. Am I right? Why should we eat rice? Why should we eat rice? Why should we eat wheat? Why should we eat vegetables? So that's the important thing that you need to know. So when it comes to the drug discovery point of view, please don't bring in the natural products here. Please. Okay? Because that's what I'm saying is my own experience. Okay? Especially you people when you go to the abroad. Okay? Go to the next one. Okay, and these are the ones. Is the molecule likely to be orally absorbed? Could it reach CNS metastasis? Is metabolism risk obvious? Is it chemical friendly? Does it improve on the reference drug? These are the questions that we need to do. And of course, these are the things that we need to look at those things. And this is the decision that we have to make. Next one. Then, of course, all these tools we talked about gene cards. We talked about PubMed, we talked about string, we talked about AlphaFold, we talked about the PDB, we talked about Campbell, we talked about PubChem, we talked about Swiss similarity. Of course, we also on and off I talked about when I say ChatGPT, I'm not saying ChatGPT, it could be any generative AI. Okay. So if you put all these things together, you can do a fantastic research. You can do a fantastic drug discovery research. Okay. Then next, is it the last? Okay. Can you open that CCA? Same. Can you go up? Yeah, here there is a Swiss ADME Euclid system. Okay, now you go down here. Okay, actually here you can draw the structure, or you can completely put a smiles here, and you can do lots of. You can put in each smile. You can put it on a single line. Can you go down? Go down. Okay. Then of course, yeah. Once you do this, actually you will get. You put this on fill with an example or see whether it is working. Something just to run it. So actually it is going to run here. These are the things that you will get. You see how nicely they put entire data into here. Actually, you can download this as a PDF file. Okay. Now I'm talking about the boiled gamma, right? This is one that you need to see. Okay. So this is the favorable region. This is the outside region. So you need to look at the entire optimization part. Okay. Go down. You will get all the physicochemical properties. Okay. Here you talk about the lipophilicity, and here you talk about the drug likeness. Okay. Go down. It also talk about some kind of a pharmacokinetics. Go on. It talks about some kind of a water solubility. Everything is there, and actually you can download. Okay. So that is the but here afterwards, once you do this, you put all those things because for you, how many things are there, madam? Here, can you go up little bit small, up, little bit up. Okay, here ibuprofen is there. Go down, madam. Go down. Omeprazole is there. Go down. Molecule 3 is there. Then molecule 4 is there. Then, am I right? There are 4 things. So for you to really do this one, if you want to do a manual comparison, it takes some time for you. There you can use ChatGPT. Hey, I want to see, can you analyze this entire, all these 6 properties? Can you rank me based upon the optimum properties 1, 2, 3, 4? It will give you. So, but if you use it for generative AI for this purpose, you are gone case. But there at that point you should use generative AI for interpretation. Okay, but can you go here? You can put this show point. Yeah. So this is the region that they are talking about. It exactly looks like an egg. It exactly looks like an egg. Okay? So that is the thing that you need to talk about. Okay, this one shows the blood-brain barrier. This is the PGP one. This is the PGP plus. All those things will come. Structure and you put here. Yeah. Molecule 3. You put it here. Omeprazole here. You put it here. So definitely this molecule is out. Okay, this molecule is out. Something like that. Okay. So that is about Swiss ADME. Okay. Then it goes to the last one is how we can use of generative AI. That is going to be my last talk. Okay. Here, yeah. Of course, here why the generative AI is important that we need to know. Okay, and please make sure that use generative AI as a research assistant. That's the take-home message. Okay, but unfortunately, what happens is we make generative AI as a master or as a professor. That's wrong. You make generative AI as an assistant. That's very important. That is the main key things. Okay. And write prompts that include context, task, constraints, evidence, output format. Of course, this is what they will say. Okay. Suppose you need to say that context. That means in this case, you are saying that you are doing a EGFR drug discovery. Okay. Then you have to give the task, and your task is you want to develop a new molecules which works on a particular mutation. Okay. Then you have to give the constraints. Then you have to say that I want you to give me the molecule of size maybe around 300 to 400, and which hydrogen bond donors containing 3 or 4 hydrogen bond acceptors are so and so. Okay, constraints. Now evidence. Now what kind of evidence you want? So I will say that, okay, please give me the chemicals that are reported in the literature, or you can also say that I want only those compounds that are reported in Journal of Medicinal Chemistry, or you can say that the compounds that are there in the ChemBell. Okay. Then you can also say the output format. In the output format, you say that whether you want it to be in a tabular form, or you want it to be in a, you want to draw it's a chemical structure, but don't do that, because so far I did not see any generative AI which can give you the perfect chemical structure. Okay. I did not come across so far. So, but you can ask with smiles. It may give the correct smiles pattern. It might give you the correct smiles pattern. And also you say that I want you to get this smiles pattern only from PubChem. Okay. So nowadays, what is happening is, because we need to write the correct prompt. Somehow nowadays the people don't even doing that. Even they don't have even that much time even to write a prompt. That's why now they call it as a prompt engineering. Okay. Of course, in ChatGPT there is something called as a prompt, but to me, my own experience, actually the prompt that I have written on my own always works better than what the prompt generator did for me so far in my case. So all these things you need to know. And again, I say that don't try to explore all the possible AI tools that are there in the market. Anyway, it's not your job. Your job is to do something research. So you stick to one. Maybe first you play around with few things. Then you say, of course, most of the things, okay, there are free tools are there and subscription tools are there. Okay. Then we need to know what is the difference between a subscription and a free tool. Subscription means, okay, of course, there are different kinds of a subscription. Whatever the data that you put in there, it is not going to be used for the training purpose, or it is strictly private and confidential. And ChatGPT is not going to, when I say ChatGPT, it's not a ChatGPT, please. Okay, it is not going to go into the public domain. That is the advantage of having a paid version. Okay, that is the advantage. When you do a free version, am I right? Just imagine if the free version gives the fantastic results, who is going to buy the paid version? It's a simple logic. It's as simple as that. Okay, but somehow the freeware versions, based on my things, fantastically looks cosmetically fast. Cosmetically looks fast. These are the general things that we need to see. So again, my own experience, I say that is the way I scold my PhD student. The same way also I scold my research assistant, that is generative AI. Okay. I asked that today I'm stressed, can you sing me a song? It sings a song for me. Okay. And if I say that this particular concept I could not able to understand, can you convert it into a song for me? It will convert into a song for me. And sometimes when I'm very tired, okay, I don't know what English you are talking about. Can you speak me in Telugu? It will try its level, but Telugu is much tougher to understand when it speaks about it. And now actually it understands me more. That's why now also it reminds me, until you are supposed to 8 o'clock, you are supposed to go to the office. Why you did not go to the office today? So it's like an assistant to me. So it understands, because actually, do you know that when you use this one, I don't know how many of you are using iPhone. Okay, there is something called Siri. Whatever I do, actually it will record it. Okay, if you install Google, actually you will know, you will trace wherever you go. Okay, even you doesn't know all those things. Actually, that means in one way we are not sure whether do we have privacy. Okay, there is no such thing. So that's why what happens is, when you take a research student, a professor, first 6 months, 8 months, 1 year, it may not stick. Am I right? Then afterwards, the supervisor understands the PhD student better. PhD student understands the supervisor better. Am I right? Research assistant is also the same. But you need to travel with that for some period of time. Then it will try to adjust. When you are putting more, of course, all these things will happen, but you have a paid version. Then it will understand what type of questions that you ask, what kind of a mood that you have, okay, all those things. Okay, so it makes your life easy. Okay, that's why stick to one thing which is good for you, which is good for your research, but your research question must be very clear. Okay? So yeah, because that's the reason why I say that when someone asks me which generative AI is good, usually of course our policy is actually we cannot promote any kind of a product on that matter. That is our university's policy. So one thing is trust one, begin a journey, continue journey for some time. If you are not happy with it, then you make a decision. Without starting a beginning a journey, if you keep on jumping from one to another, there is no point. That means it will take a longer time for you. Okay, next one. Of course, this is a high value early users. Summarize verified papers. That means you download all the PDF. You don't have time to read, then you can ask it. Okay, can you tell me why this guy did this work? I cannot understand anything what this guy did. Can you tell me in simple terms? It will tell in a very simple terms, very, very simple terms. Sometimes I say, hey, you fool, I don't even understand what you are saying. I don't know even ABCs of it. Can you tell me in a layman's language? It can go down. Okay, that means the way that I show my what is it called anger on my PhD student, the same way also I show to my research assistant that it understands me how frustrated I am. So it will give the correct answer. Okay, so that is one. Okay, draft target rationale. That means you got all this thing, all these things. Am I right? You say that you collect some data and you put everything. Hey, I got 400 pages of the documentation, but I want you to tell me, summarize it in 2 pages in a bullet format. And this is my question. This is what I want to know. It will tell. Okay, that is one that you should use. And explain structure activity relationship. Okay, what you can do is you can put a smiles pattern. Okay, in a tabular form in Excel sheet, and you can put a bioactivity of it. Hey, because we can do of course later on you need to compare with them. Can you tell me why this guy used bromine? Why this guy used the bromine? Put the bromine into it. I could not able to understand. Okay. Sometimes we are very difficult to understand chlorine, bromine, iodine. Am I right? Looks very similar. We can't. Okay. In the case of nanoparticles, you can use manganese, you can zinc, you can use iron, you can use gold, you can use silver. Then you say, hey, I'm not understanding why the people are using manganese over this one. Of course, from our drug discovery perspective, we use for gold for cardiovascular diseases, silver we use for infectious diseases, and if we use manganese for anything that is related to the magnesium, mainly manganese, for brain type things. Okay, because there are some because this very simple example. Okay, we should know sometimes the culture also gives us some kind of a hints. Okay, in culture we say gold, the women will wear gold. Okay, of course nowadays it become a fashion. But if you do it exactly, when they put the necklace and all those things, usually the size is exactly falling onto the top. That entire the rocket. Okay, because it has some kind of a power. The gold has some kind of a power. Indeed, that's why when they have started, okay, why they have chosen only the gold? Why not they did not choose any other metal? If you ask that kind of a simple question, you will understand. That is called as ancient wisdom. Okay, so from that actually we can get lots of things. Even when you go to the temple, they use a copper. Am I right? So when you go and put that one, he will put onto us and he will touch it. Once you touch it, actually you will see that you will be completely feel very relaxed. You completely feel very relaxed because it will take over all your electricity into it. And nowadays they are saying that use a copper jar, okay, put water into it and you drink it, it is useful for diabetes. Then anyone is working on the diabetes, they should develop the copper nanoparticles. That's that means everything, if you look at it from research direction, you will get. I think actually Indian culture is very rich in drug discovery. But each and everything we need to understand, we need to put a question why we are wearing only gold, not any other metal. If you say costly, titanium is more expensive. Why can't we use titanium? Okay, so or platinum, sorry, platinum. So that is those things we can do it. These are the higher risk claims. Give me 10 references for me if I want to write a paper on this. Very high risk. Okay, or can you give me some PubChem IDs? Very risk. Okay, and can you give me for what clinical conditions it is useful? Very risky. Okay, here you see use AI for speed. That's important thing. Use databases for the truth and use experts for decisions. That's all. And as long as you do this principle, these three principles, that is called as responsible and ethical use of AI. Okay, here you say, good, using these verified EGFR notes, that means you collected some notes on the EGFR, draft a plain language explanation of exon 19 deletion and L858R. That means you collected this information and you are giving input to it. Then you are asking that AI to help you to understand better. Okay, that's what you do. Risky. Tell me the exact IC50 and cite 3 papers. It's very risky. Better option is you can provide the Campbell values and ask AI to explain what they mean. Sometimes we don't understand. Then verify. You need to verify the explanation. Next one. Okay, these are the some of the things that I put, but there are lots of things. If I want to write down, I cannot write anything. But this is what I do is what most of the people who are involved in science started seeing that it is good. Okay, so this is the general large language models. This is such grounded artificial intelligence. Okay, then of course here you can see Gemini with search. Okay, nowadays when you type in Google, first thing that will come is AI, what the AI says. All right, actually then you can interact with it. We are so lucky, Gemini is free now. Gemini is free. So when you click anything, first thing will come is the AI. Then if you what is that, expand that this one, actually you can ask more and more questions with it. Then it will give you more and more answers. Okay, that is one thing. Of course, ChatGPT with browsing means what happens is when you use a ChatGPT, what happens is there are different versions are available. Okay, so if you have the paid version, it will also look at the latest literature. Okay, that means the one that is reported. Suppose if you are checking at now, what is the time at 11:30, and if you use the paid version, if someone reports at 11:20, that also will capture it. That is the ChatGPT paid version, or any paid version will do that. Most of the paid versions do that. But when we do these normal versions, they already have the data. Am I right? Then today someone has invented, it goes into the data, it will take some time. So we may miss something. So that's why different versions are there. So you need to see that. Of course, elicit, consensus, S-site, and all those things. And of course, this is institutional retrieval augmentation tools. That means institution itself will develop. What does it mean? Means, let us say you take the Raghu College. Whatever the research is being carried out, it is going to be put it into one kind of a database. Okay, then you use that as a training model and try to generate a new hypothesis. So that is what big universities and especially the drug discovery, what they do is, this is what they do. They develop their own platforms. That's why in drug discovery, never ever trust the other ones. Okay, you can develop your own model, and only thing that you require is Python, okay, Excel, and R. That's all, only these three. Then you can do wonders. Okay, and if at all, if you are doing like intellectual property rights, all those you can do it. It's an institutional property, institutional preference. And these are the molecular generative tools. These are the very specialized tools, okay, to marking about the chemistry in a drug discovery. Okay, and it's not like a generative AI. It's not exactly like a generative AI, but these are the deep learning model. Okay, here, do not ask which AI tool is best. Okay, ask what research decision am I trying to support and what kind of evidence is required. That means, suppose if you have a 10 platforms, you can give a simple test to it. Oh, okay, I want to have the EGFR clinically approved inhibitors in the market. Okay, give something. Go and check the literature. Then you will check which one is giving the correct answer, which one is not giving the correct answer. Okay, in that one also you can filter it out. Okay, next one. Okay, useful and realistic. Explain complex mechanisms for beginners. That one you can do it, because even when you give a complex picture into it, actually it will explain everything. Okay, because nowadays what I do is, most of the papers they have the figures. I could not able to understand. Just I copy and paste. Can you please explain for me? It will explain for me. Okay, what I do is, sometimes I say that when I write, my student will come and say that come up with a lot of material. Then I say, can you convert into an image? Sir, very difficult. Then I say, or put it to the my assistant. Can you convert into a proper image which can be understood, which can be understood by the researchers outside? Then it will do it. Okay, rather than please generate an image for me, you cannot. Okay, when the student writes that, when you put that, you will get it. Okay, then suggest hypothesis and search terms. Okay, or it can ask you the draft tables, summaries, and checklists. It will do for you. Suggest the hypothesis and search terms. Research hypothesis it can do. It can also translate the technical content to whatever the language that you want, whatever the simplicity that you want, you can do it. Okay, that one. And of course, for different audiences, I told you that different people will have a different ways of learning. It can help you. Help compare multiple verified outputs. Okay, that means, let us say, for example, one says that curcumin IC50 value is 10, another one say 20, another one say 30, another one say 40. You are getting confused. Then it's very difficult for you to say. Then I will say that, okay, can you compare all these 4 papers? Ideally, the experimental protocol should be like this. Okay, for example, when we prepare a nanoparticles, whatever the people say, when we do it, we are not getting it. Always a big problem. Always a big problem. Whenever we are trying to do nanoparticle, we are not at all getting a nano. Okay, there are lots of issues we have been facing. Okay, so but in that case, actually, what happens is, I will tell you one story. Whereas my student is supposed to do the nanoparticles. Okay, so that girl, she repeated 20 times. Then I said that you are wasting my chemicals. Okay, you are wasting my time. Everything you are supposed to graduate, you are wasting my time. So what I did was, whatever the notebook that she has written, I took the photocopy of it. I asked my research assistant, can you help me where this person is doing wrong? Then it rightly pointed out. It rightly pointed out that stirring technique is wrong. Okay, because studying we can do this one, or we can also do this filling. And she is consistently doing something which is mistake, okay, which we could not able to understand unless otherwise when they put that laboratory notes into it. That's why in the laboratory notes you write everything, and it helps a lot. It really helps a lot, because that's how we should use a research assistant. Am I right? And then not safe. Find out the IC50, write about clinical claims. Never ever use generative AI to claim something. Don't claim anything. Please don't claim. Research assistant, it is only for you. Whatever the output that the generative AI gives, it's to you, not to the others. Always remember that principle. Okay? Then you go and read. Yes, you can use it, but before you tell to someone, you read it first. Whatever you are saying, does it make sense? Okay? Then don't talk about safety decisions and all those things. Use AI to accelerate thinking. Okay? Use primary evidence to confirm the facts. That's the most important thing. Next. These are the pros, okay? Of a strong natural language explanation, good for drafting the research proposal. Yes, it's really fantastic. And there is a UK, very recently, I told you that I'm on my way from UK to Malaysia here. So there they're talking about UKRI, that is UK Research Initiative. Now there is about 2 days of debate whether we can use generative AI to come up with the research proposals. Now there is a lot of debate that was happened when the generative AI is there. Everything comes very good. Am I right? Everyone writes a proposal, good. Then at that time, I told my this thing, but the people say that still they are not going to accept it, but in future they are going to accept it. Okay. Then I say that yes, any person can say anything. I can do that. I can do this. I can do this. But it should be supported by the CV. We should be supported by the papers. So my view is we should encourage the people to use the generative AI either to write papers or to write research proposals. But however, it should be validated and it should be supported by the CV and the publications. Am I right? So the person can lie in the proposal but cannot lie in the CV or the publications. So that means what I'm trying to say is in future, I think generative AI is going to play a big role. So better to be alert, but there are 2 things: ethical and responsible. Those 2 are the most important. Okay, next one. Of course, these are the various tools that I put. What is the best tool that we can do? What is the strength and what is the limitation? And even though I put this one, okay, use literature AI to find and organize papers. Very good for organizing the papers. Okay, sometimes we do not determine the quality. Okay, suppose if I want to write a literature review, okay, I will collect suppose 10 papers or 100 papers. So I will say, can you tell me, can you only please pick the papers which are published by an institution whose QS ranking is between 1 to 10 or 1 to 20 or 1 to 30? It will give you. Okay, or you can say that I want you to extract the information which is published only in Q1 journals. It will give you. Or you say that please give me the papers that are published in ACS. It will give you. Okay. And then you say that okay, I got so many 10 papers, but can you evaluate the experimental quality of the experiment what they did? Then you do. So here, if you see carefully, the reason thought is coming from here. So I'm asking a correct prompt because I know what I want. Okay. Yes, I can do it on my own, but it may take days. But when I use my research assistant, it makes my life much easier. That's all. That is the use of the generative AI. It's not the okay. So then focus on the peer-reviewed literature search, and of course SCite and PubMed AI and all those things you need to do. But as I told you that sometimes the people are also creating references using generative AI that is considered as unethical. Unethical. Never ever generate references. But you can take the help of the I want ACS patterns. Yes. Then you go and check. Okay, please. Okay. Use literature AI to find and organize paper. Use PubMed or full text to verify methods, endpoints, populations, essays, and conclusions. Now my journey with my research assistant, when I started my journey with that, I was not so happy with it, but scolded. Now it is giving me very good output, which I like it. Okay, don't since you cannot say that, oh, since you are saying that particular research assistant is working for me, why can't you tell me that research assistant I also use it? No, because I traveled with it. So it understands me very well. Really, it understands me. When I say that, it really, really understands me very well. Okay. So that's why it works. Next one. All these things, reinvent. All these are the more towards chemistry ones. These are not like a free ones, and all these things mostly are published in ACS journals, American Chemical Society journals, and lot of research is going on actually to do this. Okay? And generated molecules are hypotheses. This is very important. And because sometimes, okay, even I also have seen few things. AI generated some papers, some compounds. AI has generated NMR predictions. AI has generated possible activities, everything written a PhD thesis in one day. Can is not at all, because everything is fantastic, am I right? It can even generate NMR spectra. Everything it will do it. Okay. What it cannot do is, when you do the NMR spectra, there is a parameter. That's what it cannot generate. Once if you are good, then automatically you will know that whether it is a fake data or it is generated. That's why now I don't accept any NMR data without the parameters that are being used. Right? On the left hand side, there is a parameter to be used. Right? So for all the experimental methods, what I see is I want to see the experimental conditions that are being reflected on that paper. So that is one thing. So these things are happening. The people are using it, but there is no use for it. There is no use for it. Okay. Next one. Okay. Of course, I told you that context, task, evidence, format, and border, everything is this one. A strong prompt reduces ambiguity but does not remove the need for verification. That means whatever it gives, you need to really verify it. Tell the model what you know. Okay. So for this one, you need to have the subject. Then only you can tell. Okay. Then you need to have your aim, what you need. You tell that. Then and later on you see how you will verify it. Next. So here, okay, let us say, tell me about EGFR drugs. Nowadays, all the people are like this. They don't have even energy. But to me, if you write this one, using the following verified notes, again, verified by us, okay, create a beginner-friendly table comparing the EGFR inhibitors by generation. That means whether it belongs to the first generation, second generation, or third generation, mechanism, resistance mutation address, and a clinical relevance. Mark any claim that needs me to verify. That means it is not confident. Then it says that, hey, Arjun, please go and check on your one and beat this one. When you do this one, because if this prompt may take maybe 2 minutes to write. So if you do not have this prompt, actually to do the same thing, for you it will take 1 week. But it is reducing a lot of your time. Okay, that is one thing. Okay. Of course, whatever the numerical values that it use, don't believe that. Any numerical value, don't believe it. Don't believe any reference. All those things, citations, those things, don't believe it. Okay. Better prompts create outputs that are easier to check. Okay. Otherwise, because if you give a very bad prompt, am I right? It will give lots of info. Then it's a headache for you to really check. So it won't save your time. So please, next one. Here, these are the common mistake. They ask broad questions. Okay. Simply say whatever it says, we believe it. And we accept the numerical values. That is the common mistake we do. And we are ignoring the confidentiality. Okay. There are a few things that we do. And no record of prompts. Actually, people don't write down somewhere what prompt did I use. Okay. And in future, I am expecting that the journals may ask, please share your AI prompt that you use to generate the text. I think that is the best way to go forward, because we cannot stop asking, we cannot stop the people using AI. We should make use of the technology, but we have to be responsible and ethical in that aspect. Using, as I told you, this is my favorite ones. Okay. A good AI is transparent, reproducible, and source grounded. Next. This is what signs of hallucination. It will do it. Even we also do it. When our brain is very tired, what we will do? We can't talk properly. Okay, we also hallucinate. So, references that cannot be found, optimal lines do not match with the claim, very precise values. It's highly impossible. They will say 10.05678 something. Huh? Because how is it possible? Am I right? Any paper they will give a 2 decimal points maximum. Am I right? So that thing we need to see. Overconfident answer to an uncertain question. That's very important. It tells very confidently. That means okay, you should not trust it. Okay, because you know that that is very ambiguous. Okay, conflicting of answers across different prompts. Okay, how you can do is how you can reduce your reduction or reduce is you give your data first, then ask for uncertainty and assumptions. Require source specific verification steps. That means you need to have all the prompts. Use retrieval or source grounded tools that when appropriate and cross check primary data always. That means no need to check all. Right? Check on and off. Okay? Maybe random pick and check whether it's okay or not. Okay, this one. If AI states PDB 3LZB proves this particular drug binding to the this particular mutation, don't accept it. Don't really accept it as it is. You go and check. Because this is very positive, it is telling very confidently, highly impossible, highly impossible. Okay, then when it is telling so confidently, don't accept. Okay, hallucination risk is manageable. We can manage it only when verification is built into our workflow. Next, I think this is the last. Okay, these are the useful questions that you can ask. What evidence types support this one? All these things you can use it. Okay, a target is validated by evidence, not by a convincing AI explanation. Next. Okay, that's all. Thanks for your time. Thanks for your patience. Okay, I truly appreciate your time. Thank you. I hope you got something from this workshop. Okay. Thank you. Bye.
Thank you, sir. Thank you very much for your valuable insights into this session. Now, I request any one of the participants to provide their valuable experience on this 3 days workshop. I request anyone from your participants. A very good morning to one and all. We are really overwhelmed with this 3 days FDV, which is organized from our Raghu Engineering College and Department of Pharmacy. So really, it was a great experience to be here all these 3 days. We have learned a lot of things, and I think everyone got so many inputs regarding AI. Starting from the beginning, we have understood the AI technique, how can we use it, and we are really fortunate to have Professor Mallikarjun sir as a resource person for this workshop. And he has taught us everything starting from the beginning. How can we use AI for research, for teaching, not only for research as a, I think he's an excellent teacher. Everyone should agree with us. And he taught us a really great thing. How can we use AI and make AI our assistant? And whatever task we give, so that we can get the task to be done immediately, which will reduce our work, and I hope that everyone is going to use it in future and effectively, and you will get benefit of it. So thank you very much, Sir Mallikarjun sir and Panda sir, for giving us this opportunity conducting this such kind of workshop. In future, I will expect from sir to conduct like that many more workshops, and we'll also like to have Mallikarjun sir again in future if possible to enlighten us with his vast experience. So thank you very much. It was really a great experience overall. Thank you. Thank you, Dr. Anandita Madam, for your feedback. Now I request one of our students, any one of our students, to provide their feedback.
A very good morning to one and all, our respected dignitaries and our chief guest, our Mallikarjun sir, and our principal sir and our dear faculty and all the students who are present here. This is Surya Kumari, pursuing my Masters of Pharmacy in Pharmaceutical Analysis branch in Raghu College of Pharmacy. This 3 days workshop has been a brainstorming thing for all of us, and we are deeply thankful for for our guest speaker sir for sparing his time to come in here and sharing us deepful thoughts about the different AI tools that can be used during the drug discovery. All the tools that have been discussed in the 3 day session, which are all useful, which includes Campbell, PubChem, Spring, all the tools we can use for our future self. So we have learned so much things from these, which we can implement further in our future. We can participate in research programs if we want, and these have been a great session for us. And the session which you have conducted for us has spiked our curiosity making us to learn more about them and use them in our daily life. Thank you, sir, for all your valuable input you have given us through this session. Thank you so much.
Good morning, respected principal sir, teachers, faculty, and all my dear friends and all the guests. Thank you. I sincerely thank Mallikarjun sir for giving a wonderful lecture on AI tools and for this 3-day workshop. Everyone got understood about these AI tools and their influence in further future. So I request everyone to use these AI tools for who are pursuing B Farm level. Thank you, sir. We use this AI tools in 4.1 and 4.2 projects and practice school. Thank you.
Thank you, students, for providing your valuable feedback. Now let's proceed to a valedictory function. So before proceeding that, I would like to request the dignitaries onto the dais. So let's welcome our principals with a round of applause. Now may I request the principal sir to greet and welcome our chief guest of the today's function, Dr. P. Mallikarjun sir, onto the dais. Thank you, sir, for gracing the dais. So with deep sincerity, I would now like to request our principal sir to speak some of the words from his concerns regarding this workshop.
Audible? Yeah, first of all, I would like to thanks Dr. Mallikarjun to have with us for these 3 days and extensive lecture regarding this artificial intelligence in drug discovery and drug development. With taking a case study of the language. First of all, the journey was as I gave in a webinar, I gave one lecture, which made me learning myself and teaching something. And on that day, as per the assignment I have done, but I felt that a person who really practice and who is practicing, so if such person if take a workshop in this region, the teachers and the students will enlighten with the knowledge, and definitely some of them will get ignited in the future prospects, in their future research. They will engage in the drug discovery process, and that will be beneficiary for the society. With this philanthropic thoughts, I've been searching in the, web sources, YouTube and different sources. So I got so many ideas, and out of that, and happens that Dr. Mallikarjun, who is one of the very good researchers, is a dean research of the Newcastle University, UK, which is Malaysia based, and he's being my classmate and my friend. And as I came to know that he's coming to the India, I requested him, and it was very impossible to stay with us for 2.5 days, 3 days. But even then, as I told the truly what my feeling was, then he accepted the invitation, and this is the what was the contest. And sincerely, I thanks for this, sir, for your accepting me. And as for that one, you know that he have been delivering the all these 3 days in spite of his busy schedules and a lot of mails and all. So he done his job 100%, where he tried his level best. And after that, actually, what was my dream was that to learn everything. I understood that after these extensive lectures, we could learn only the basics only. What was my dream was, how the generative AI will make the generate the new molecules of taking some example of the some disease, from disease to the some proteins or enzymes, whatever it may be. And that is target validation, and hit lead affirmation validation, and after that one, retrosynthetic analysis. So that was my dream. But I understood that even to make them understand what is the chemical space and what are the different databases are available to understand the chemical space, that itself took yesterday and the time. But anyhow, these basic points definitely make you some question marks in your mind, and thought process will generate. And definitely in future, out of you, at least 5 members come as like him. That would be a great to this nation. And first of all, I take these young two boys, researchers from Andhra University, they come, and appreciate them for their interest. Definitely, you will blessed with this knowledge, and that will be helpful for you and other students also. I thanks to the all the students and the online and offline who participated in this one. That was actually what my journey to conduct this one workshop, and what was my feeling. I told very clearly. And after that, you all supported. I very much thankful to you, and especially my crew, especially Dr. Swati Putta, with whom I started, and later on Harini, later on Nilima, and Gauri, who did a lot of job on one day crucial time, entire the our generating the what do you call brochure and all, and later on the a great very strong fellow in our college, technically very sound and strong, Anita Panigrahi and Nilima, they have taken the lot of pain and a lot of exercises, okay, and permutation combinations to make the live very systematically, because this is the streaming into the YouTube where the entire world is going to watch. That I am very much aware, and that's why I've given a lot of instruction. As per the instruction, very patiently they followed. And these three young boys here, we have seen that they've done a very good, wonderful, commendable job. So in making this actually very true successful. Every step will start with a single step only. Every version will start with a one drop. Okay. Similarly, this is my sincere, honest approach to make the some curiosity, some level of thought process in the young minds and the teachers research towards the drug discovery and recent trends in the drug discovery. And as I'm myself very much fashionable in AI tools, every day almost I spend 1 or 2 hours for AI tools, and so this is the appropriate time what I felt, and that's why we have done this. And this is what my entire journey of myself, my thought process, and my supporting with Dr. Mallikarjun. And other, he has a very important point that this kind of the, all facilities, it is not a possible for any pharmacy college. It is the possible because of Raghu Engineering College, especially the strong support of the our chairman, sir, K. Raghu sir, and without whose help it cannot be possible. With his kind permission, we could do this one. And later on, very much we have to commendably to say the name that is Dr. A. Vijay Kumar, the principal of Raghu Engineering College, and whose sustained support to make this is very much successful. Similarly, the head of the department, Srinath Raju, Dr. Srinath Raju also gave the wonderful support for this. And from that, the same time, I thought that AI, ML is, if suppose a computer science fellow teach the basics, that would be better, I felt. Then Asima Naik came and she gave the first lecture. And later on, I thought that now recently we are going to purchase a software and which was made by the Indians, okay, and that is called the Rasayana, okay. So Rasayana Limited. So I asked them because they are made a one very good software where they do all the things like from target validation, target identification validation to till the electrosynthetic analysis part, all the drug discovery part they are doing in their software. That is possible. So I requested them, and readily they accepted that. And one of the person who's really doing the synthesis using the software from IRCT, Mr. Singh, actually he came to the take the delivery lecture and wonderfully made, and all of you witnessed that one. Okay. From since then, Dr. Mallikarjuna is taking the entire sessions, and he gave a wonderful lecture. And this is what actually, yeah. So this is the what entire my story of this one. And during this, I have to mention here some of the support that Vigyan College principal, Dr. Y. Srinivas, he gave me very good support, having the good registrations from the student as well as the faculty. Of course, because of and also all the colleges they have participated. Other colleges also they in and around they have participated because of their times busy schedules. Okay, they could not attend. You know, offline, but almost every college they participated, around 50 members delegates, because we are limited for only 40 to 50 members only. So they have joined in this offline and online. And first of all, a noteworthy, Dr. Padi is from the professor from Central University, Raigada campus, Gopal Padi. He came today and he has been following all the classes, lectures yesterday and before yesterday. And similarly, Mr. Sarosh Kumar, Dr. Sarosh Kumar, also from the Vijayanagar MR College of Pharmacy, he also witnessed today the program. And first of all, thank you. And other my student and the principal of Vijay College of Pharmacy, and Mr. Madam Nagarani, Dr. Nagarani, is a very well-known pharmacology scientist. She also participated in this. And other my fellow colleagues from MSc background from Raghu Engineering College, my colleagues have joined, and other madam from the computer science also joined, chemistry department also they joined, and first of all they and other my all colleagues, and thank you one and all for making this program very successful. My aim is that to create certain awareness and make the interest in the thought process of the younger generations and the younger teachers who are doing in the research in this field. And thank you one and all, and Harini and team for their commendable job. And thank you all my colleagues Rama Devi and Padmaja and Sati Paul, all are involved in the program. All my other my non-teaching staff also they gave the lot of service to one and all. For everyone, okay, that Padmaja, that Rama Devi, for everyone, I must thanks to everyone making this very successful. All my colleagues, for everyone, and I might have forgotten maybe because of this time. Okay, thank you one and all, and I request for further questions.
Thank you, sir. Thank you for your remarks. So, with this brief remarks of our principal, sir, now may request our principal, sir, with his team to provide a token of love to our guest speaker, Dr. P. Mallikarjun, sir. So, now I request all the senior faculty to accompany our principal, sir. Thank you. Thank you, sir. Thank you, sir. Now I request Dr. G. Gauri, Associate Professor, Rao College of Pharmacy, to propose the formal vote of thanks.
Good afternoon, everyone. Myself, Dr. G. Gauri, Associate Professor, Rao College of Pharmacy. It's my great pleasure to propose vote of thanks at the conclusion of this enriching three-day workshop on artificial intelligence in drug discovery and development, a lung cancer case study. On behalf of all the participants, I express our sincere gratitude to the Kalidindi Raghu sir, Chairman Raghu Educational Institutions, and Dr. Jagdish Panda sir, Principal Raghu College of Pharmacy, for their continuous encouragement and support for this well-structured workshop. I'd like to thank Dr. A. Vijay Kumar sir, Principal Raghu Engineering College, and Dr. Srinath Raju garu, HOD Computer Sciences Raghu Engineering College, for their support and providing all the facilities for this workshop. We extend our heartfelt thanks to all the distinguished speakers, Dr. Anima Naik ma'am, Professor Computer Science Engineering, Raghu Engineering College, for introducing us to artificial intelligence, machine learning, and deep learning in drug discovery. Mr. Ranbir Singh Saluja sir, research scientist, Rasayan AI Lab Private Limited, for explaining the AI applications across the drug discovery and development pipeline. And a special note of appreciation to Dr. P. Mallikarjun Rao sir, Professor, Research Dean, Newcastle University, Malaysia campus, whose expert guidance throughout the workshop and hands-on demonstrations on gene cards, string, AlphaFold, Campbell, Pupken, and PDB, Swiss Similarity, Swiss ADME, and ChatGPT greatly enhanced our practical understanding. We also express our gratitude to Dr. K.V. Ramana sir, President APTA, AP State Branch, for the continued support and guidance for this workshop. We also thank the organizing committee, coordinators, technical support team, and volunteers for their meticulous planning and seamless coordination and constant support that made this workshop a grand success. We extend our thanks to Dr. Y. Srinivas Rao sir, Principal, Vignan College of Pharmaceutical Sciences, for supporting this session, and to all the participants who came offline and online, especially AU students and Vignan students. Thank you for your enthusiastic participation, thoughtful discussions, and active involvement throughout these 3 days. Your curiosity and engagement made every session interactive and meaningful. We sincerely appreciate everyone who contributed directly or indirectly to the successful completion of this workshop. With gratitude in our hearts and new knowledge to apply in our future research and careers, we conclude this workshop. Thank you once again to everyone. Thank you and have a wonderful day.
With this, we come to the end to the session of the 3-day FDP on artificial intelligence in drug discovery and development. Hope every participant has come to a conclusion that there is more to do in the field of drug discovery using artificial intelligence, and we bring more sessions in this same format in the coming future. Thank you everyone for attending this FDP. Myself, Antarash Panigrahi, Associate Professor, Rao College of Pharmacy, signing off. Thank you, everyone. We are ending the stream.